of enzyme action. Today we will look at how enzymes and substrates react with one another to form products, the kind of mechanism involved. Before that, let us get to know certain basic aspects. The enzyme is always considered as a protein molecule. We all know this. In addition to this, the molecule that enzyme works on is referred to as a substrate. Next, we see the produce from the reaction of the enzyme and substrate is referred to as a product. And the part of the enzyme into which the substrate has to fit into is called as an active site. We will learn more details on this. Therefore, enzyme action has the following prerequisites. We need to have an active site of an enzyme. The mechanism of enzyme action has to be determined, and the specificity of the enzyme action plays a very important role in the formation of the right kind of product. So let's begin. Every enzyme has a part on itself which is called as an active site. This active site is the region of the enzyme that binds specifically the substrate. So as you can see, the enzyme binds the substrate on only the active site. When this happens, an enzyme substrate complex is formed. This is the first step of the reaction whenever an enzyme reacts with the substrate before releasing the products. So therefore, we can see in this slide the substrates binding to the active sites on the enzymes, forming an enzyme substrate complex, and after which the products are released from the enzyme substrate complex. To further make it clear, the mechanism of enzyme action is shown here, where you can see a molecule A which can be considered as a substrate. The green area that you are seeing as depicted as the enzyme shows also the active site where the molecule A, that is the substrate, fits in. So this is the active site. So therefore, the process of catalysis happens to produce the enzyme product complex, after which the enzyme releases the product, and this enzyme can be used again. In an enzyme catalyzed reaction, a substrate attaches to the active site. Then an enzyme substrate complex, that is ES, is formed. Reaction occurs and the products are released. This enzyme, as mentioned earlier, can be used over and over again. E plus S, that is enzyme substrate complex, forming the enzyme's product complex and releasing the enzyme plus product. The substrate here binds to the active site of the enzyme, fitting into the active site. So we have E plus S, which induces the enzyme to alter its shape. Then the fitting actually has to take place more tightly around the substrate. Only then you can form an effective enzyme substrate complex. The active site of the enzyme breaks the chemical bonds of the substrate and the new enzyme product complex is formed. Finally, the enzyme releases the products of the reaction E plus P, and you get the free enzyme which is ready to bind to another molecule of the substrate. In addition to this, we also have a coenzyme, which is a molecule required by the enzyme for catalytic activity. A coenzyme therefore prepares the active site for the catalytic activity of the enzyme. The mechanism of enzyme action has two different kinds of methods. One is called as a lock and key model. The other is an induced fit model. In the lock and key model, in the same way that a key fits into a lock, So a substrate is thought to fit into an enzyme's active site. The enzyme is considered as the lock and the reactant is the key. So you have the enzyme plus reactant forming the enzyme reactant complex and releasing the enzyme plus the different kinds of products. It's more clear in this picture where you see the substrate in the form of a key, the enzyme depicting a lock, and then you have a substrate entering into the enzyme at the active site to form the lock and key complex. Also, you see here that it attaches itself to the active site to form the ES or the enzyme substrate complex. In the induced fit model, we see that the enzyme alters its conformational changes. It brings about conformational changes on its surface, producing the enzyme substrate complex to hold the in substrate and thereby release the products. So in the lock and key model, the shape of the substrate and the conformation of the active site are complementary to one another, like how a key exactly fits into the lock. In the induced fit model, the enzyme undergoes a conformational change upon binding to the substrate. The shape of the active site becomes complementary to the shape of the substrate only after the substrate binds to the enzyme.